And it's part 2 of the cyanobacteria detection and quantification. And I'll get straight to our first speaker because we are running a little bit behind schedule. Florence Choo from University of New South Wales here is going to present on using in situ fluorometers as real-time cyanobacteria removal validation tool for water treatment plants. Thank you, Florence.
Thank you for the introduction, Lisa. It is a mouthful, so I'll just get right into it. So I think we all know what cyanobacteria are now from yesterday and also today, but the also important things are they can fluctuate in terms of space and time, which can prove an issue, and also they're in different water depths. And we all know the hazards associated with the blooms, so I will skip through most of this. So they produce toxins, and there is this guide limit for microcystins, which is, I think, also being revised currently. And the US EPA has listed cylindrosmolepsins and microcystins as low as 300 nanograms per litre for children. And also another major issue that hasn't really been addressed a lot in this workshop is that they can produce taste and odours. Even though they're not been known to be harmful, they can be a cause of customer complaints, as people assume that if there's fishy or earthy smells in the water, that the water is unsafe. So it's a public perception issue too. So we conducted a sampling problem throughout Australia, so from Brisbane to Adelaide, as this is not a localised problem. Cyanobacterial blooms and associated metabolites can occur across the world, especially also in the US and Canada, and all throughout Australia. So during this time, I'm a PhD student, so my 3 years, we managed to visit 11 plants in Australia, 1 overseas, and there's 24 visits throughout these plants, and they all have their own challenges. So before I mention, so I know there's been a lot of work on looking at reservoirs and monitoring reservoirs, but I would like to mention that just because there might be a big bloom in a reservoir, sometimes that bloom won't actually go to the water treatment plant. So I'm focusing on what happens if that bloom breaks through to the treatment side. So initially in 2016, we did publish this review, and there are now more reviews in water research, for example. But this review highlights a problem with these fluorometers. So they are available and have been used by utilities, but I noticed the main problem with these fluorometers is that they can be impacted by the presence of other interference sources like green algae. So just to recap on how these fluorometers work, you put this fluorometer in a target water body. It sends out a wavelength at a specific wavelength, which excites the pigments in cyanobacteria, so phycocyanin in this case, and also can detect chlorophyll A, and which is then detected by the fluorometer. So if you look over here, so here we have 2 figures. These are fluorescence excitation emission matrices. So the first figure A is of a cyanobacteria, microcystis, and no green algae added to it. So we have here our phycocyanin peak, which the fluorometers are aiming to detect. And these are just fluorometers already available, already being used in utilities and where they're starting to fluoresce. So you want them to be in this region. But what happens if you use these probes on something that's not a cyanobacteria, for example, a green algae, in this case, chlorella? So this green algae have 2 peaks, so 2 chlorophyll A peaks, so 1 here and the second peak here. The white boxes represent a chlorophyll sensor, and they're detecting this chlorophyll peak, but there's also this phycocyanin sensor is detecting this second peak that can appear, and that's what's causing a lot of problems with these fluorometers. So we investigated this impact of green algae and turbidity on these probes. So we did a systematic study onto how bad this measurement error can occur. And so this is just an example of what we did. So we took all the probes together. We had different concentrations of green algae and turbidity in the suspension. So these are bench scale tests. It would be great to do it in the field, but it's very hard. And so on this axis, we have the measurement error. So this is the error shown by these different available probes, fluorometers. Would be 0. But as you can see, they're not all 0, and different fluorometers work differently. For example, this one has a huge measurement error when there's high concentrations of green algae and turbidity. So it's now been summarized in this paper, if you're interested. It's a free open access journal, so you don't have to go through a paywall. And it go through the different fluorometers available, 4 key cyanobacteria species, limit detections of these fluorometers based on those species, and it's just a evaluation of these fluorometers. So from these experiments, before we put them into the field, we had to correct this measurement error. So we developed correction factors that use chlorophyll and phycocyanin to reduce that error based on those experiments. So we were able to develop 4 different factors for the probes. So the important thing is, so these are, let me start again, the foundation for maybe perhaps future correction factors which work on a probe and cyanobacteria set. So we're able to reduce the measurement error of the probes by some degree. So we have, for example, our increasing green algae concentration and the relative error compared to the actual concentration of that sample. So the blue circles represent the uncorrected measurement error. The green represent a previously published correction error, and our red is our own correction factors, where we're trying to reduce this measurement error. So there are important things to take from this. That the first thing is that even though the previous fluorometers showed a huge measurement error, they were actually easily corrected. And then we showed that fluorometers with previously low measurement errors, they tend to be overcompensated. So trying to still work on that. And yeah, and so some fluorometers might actually work better with corrective factors and some might actually not. And also the width of the circle is actually the relative influence of turbidity. I forgot to mention that. So what does this mean for the field? So we actually apply these correction factors to 2 water treatment sites in Australia. So we have, for example, 2 plants in this scenario. We have water treatment plant 1 in South Australia and a second one along the River Murray. So for example, these are the 2 extremes of applying these correction factors, like 2 extremes examples. So we have here the different points throughout the treatment plant. So from the inlet and to clarify sludge, for example, as point of interest. And also we conducted these sampling schemes over various days. So we have the blue, which is our corrected fluorometer meeting based on the corrections, and then convert it to a cell count, and also the actual cell counts from the sampling event. So our first example works relatively well, but our second example did not really go as planned. So I can explain why this didn't work. So the first example is a reservoir, which only has a single species that usually dominates in these plants. So we're able to figure out what that species was and apply the correct correction factor to this probe. So it worked well. But the second plant, because of where it was how the water is sourced, the species changed, and they could be dominated by multiple cyanobacteria species, even including ones that we did not investigate. So the correction factor didn't work as well. But there is scope to use these probes in localized sites. So how does this relate to optimizing, using these probes for optimizing treatment? So we did more bench scale tests, but we decided to use the fluorometers before and after just jar testing. So in this study, I'll go through, we tested for different species, and we tested them based on growth phase, which hasn't really been explored before. So our idea is we have different ranges of coagulant dose and PAC dose, and we're trying to get a removal. So this is just the method we chose. We used Microcystis aeruginosa, Dolichospermum, Zosnelli, and Cylindrospermopsis raciborskii. And so these strains were known toxin producers, and we conducted these jar tests during the early exponential phase, late exponential phase, and stationary phase. And we chose these PAC, and alum dosages based on what the plant would usually put out. And so including that, we also took a range of this analysis. So I'll focus also on some of the algal organic matter data that we collected, because I think that might be of interest to some people in this room. So this is an example of what we collated from all those jar testing data. So as we increase our coagulant dose and our PAC dose, we'll get this removal determined by the fluorometer, and B, the cell count validation. So it was good to see that, for example, these graphs, we got similar removal rates between two fluorometers, and importantly, similar removal rate from the fluorometers and the cell validation. And we conducted a Pearson's test to check this correlation. And for this example, this was microcystis, which produced microcystins LR in this case, up to 20 nanograms per liter in the stationary phase. And we're able to show that some, at this alum dose, some removal was achieved as you added more PAC. So I thought that was pretty neat. So some of this algal organic data we collected from these jar tests. So what really surprised us, we chose these two species, and early exponential, and stationary phase, that we found that the concentration of some of the organic matter based on LCOCD did change before and after treatment. So we're still trying to figure out what this is. I'm not an organic matter expert, but it is interesting, and there is work going into it. But mainly, we found good removal of biopolymers, humic-like substances, and some building blocks. But the interesting thing is we saw an increase in lower molecular weighted neutrals in all the species. So what was the effect of alum on this algal organic matter? So we found that there was varied removal for some of the components. So as we increase our alum dose, this is our concentration on the y-axis of our organic matter. So we found that the red and the orange lines, there was good removal, but there was actually increase in this one, which was a bit strange. So good removals of humic-like substances, lower weighted moleculars, but no real removal of building blocks and biopolymers. Or good removal of building blocks too, biopolymers too, sorry. So this is just more of the type of data we collected. We're still going through this. And there is apparent removal limit of some components for alum. So you can add as much alum as you can. There will be a limit to some of the organics being removed. And that's what we found in these bench scale testing. So going back into the field work, I did mention that we did go sampling out to 11 treatment plants for a total of 25 visits during this period. So we collected our samples for water quality data and also used our fluorometers. So this is just an example of the data we collected. We wanted to see to make sure that whatever the fluorometer was reading sort of matched the same trends as the cell count data. So that's what we wanted to really see. So this is an example of a taste and odor event at one of these treatment plants where geosmin went up to 250 nanograms per liter, and what the probe and was reading and all the complementary data. And also another component of this this project was to actually install these probes for long-term monitoring. So we installed 3 probes at 3 different treatment plants, so 2 in South Australia and 1 in New South Wales. So the idea is to have them at the inlet monitoring the water with all the water quality, other online systems. So we're able to catch an event where microcystis cells actually broke in through the treatment plant, and we have the complementary cell data that corresponds with it. I just want to highlight the important thing about this data. So this fluorometer was able to capture this event in real time. So it was logging for 5 minutes, 5 minutes at a time. By the time you get your cell counts, the cells are already in the plant. So if operators had access to data like this, integrated into real time, they can actually see cells going in, so they can actually adjust the treatment processes accordingly to either turn on the PAC or to turn off the PAC when the event's gone. So I just thought that would show you the benefit of having these fluorometers for treatment. So just to conclude, we had found good correlation for the bench scale testing for treatment plants, and also relatively good correlation in trends for the full-scale work. But there is scope for future work for this project. So ideally, we want to integrate it into SCADA, so operators can actually have access to this real-time data. And this is a relatively young project. More data would help will help make better models. But there is scope for this to be applied for real-time coagulant and PAC dosing, and there I have some good ideas about that. So I like to acknowledge, because this is part of a linkage project, I would like to acknowledge all our sponsors, especially SA Water and Water New South Wales, for all the fieldwork they helped us with. Without this, I wouldn't have all this data, and I wouldn't be here presenting it to you. So if you have questions, feel free to ask, or you can ask my supervisors. Thank you.
Thank you, Florence. Does anyone have any questions for Florence?
I missed the very start of the talk. Under what conditions are the measurements made with the fluorometers? Are they fully dark adapted cells, or are they in a natural environment subject to fluctuating light?
So in the field, they were subject. Some of them were subjugated to the light. So it was. We tried to put them actually where they was at the plant. So if it was a sedimentation bay, we'll put them in the sedimentation bay, and it wasn't covered. But for the validation experiments in the lab scale, we did cover the fluorometers. Yeah. So if it was lab-based, we covered it, but in the field, it's hard to do it. Yeah.
So do you have a feeling for how the different brands of fluorometers deal with non-photochemical quenching?
Yes. So a part of this project, we also did rank the fluorometers based on what we felt, based on the optical designs and how well they did in terms of this interference. So some fluorometers don't handle quenching well, and we do see that for some of them. Yeah.
We got any other questions? Thank you, Florence.